Welcome, everyone, and it's a pleasure to be here. So I'm going to tell you a little bit about Sigilon. So Sigilon was founded about 2.5 years ago, and the partners at Flagship Pioneering, who are our funders, had this view of how do they create a first-in-category new modality with cell therapies that you can use to treat chronic disease. And so they thought, well, we want to restore normal cell function. We want to replace deficient proteins. We want to produce these proteins in a programmable fashion. And we also want to shield them from the immune rejection and fibrosis, which has been a challenge in the allogeneic space forever. Avoid interaction with the host genome, and then deliver an off-the-shelf product. So that was the vision. And Sigilon was created to, using technology which we believe will achieve that vision. And so what we have is we have a technology, we're calling it Shielded Living Therapeutics. And it's a proprietary product platform that overcomes some of these limitations that I just mentioned on treating chronic disease. We harness the cells and using the cell to actually become the therapeutic and produce that therapeutic. We are rapidly building our pipeline. Our first IND is targeted for end of this year with several followings shortly thereafter. And we certainly believe that chronic disease does present a very large opportunity for Sigilon. So in basics, this is the platform I'll talk about. There's two key elements to the platform. One is the cell. And we've taken a lot of time to create and engineer a novel cell which is something that we can then utilize as part of our platform. And so engineering this cell, making sure it's a cell that has all the characteristics that we need, that will be long-lived, that can divide but only when we want it to, can act a little bit like a macrophage to eat its neighbor if the neighbor dies, and create a sustainable cell inside the matrix. The matrix is the second piece, which is important. So we have a proprietary immune shielding matrix, and on the outside of our matrix, we have a library of small molecules we can use that prevent the body from recognizing these spheres as foreign, and so they can sit in the body for years. And I'll show you a little bit of data on that. And finally, these are placed inside the body where there's no need for immunosuppression, and to provide what we believe will be a functional cure for chronic disease for a long period of time. And so this is, I want to show you a little bit around the technology, because for me, I saw pictures like this and it convinced me to leave Pfizer and join Sigilon because it was quite compelling. So on the, let's see, that would be your left-hand side. So polystyrene beads, everyone knows cells love polystyrene. They love to grow on it. So on the top are polystyrene beads that have just been placed into the mouse peritoneal cavity, left there for 2 weeks. On the bottom are beads we've treated with one of our small molecules and removed after 2 weeks. You can see that after 2 weeks, there's effectively no foreign body growth or fibrotic growth on those beads compared to the control. Similarly, we can coat a different polymer, which is a PDMS polymer, and the same thing occurs. And this was after 12 weeks after sticking it into the mouse peritoneal cavity. So then if we then look at these spheres, so you can see on the far right, you've got the bottom is a sphere that's been coated with this afibromer technology. And you can see, you can't even see the sphere, and this is in where we stained for a mouse macrophage marker to look for macrophages that would adhere to the sphere if it was beginning to go down that fibrotic cascade. On the top is a capsule that has none of the afibromer. You can see it's starting to fibrose after 2 weeks. And so we've got a lot of data that clearly shows that we do not see a fibrotic effect in multiple animal models from mice up through non-human primates for many, many months. So what is this platform? What have we demonstrated? First, it's efficacious. So on the far left-hand side, you can see that what we have here, this is in a hemophilia A mouse model, and this is looking at bleeding time. And so we have demonstrated one that is efficacious. We can cure the hemophilia A mice with these capsules that contain these cells that have been engineered to produce the factor VIII protein. They're also controllable. We have you can see in that middle section, so low, medium, high are doses or number of capsules given to the animal. And you can see we can give a very low number of capsules and get up to therapeutic relevant range. And we can also give very high level and even push it up well above what you would expect, normal range being about 75 to 150% on this chart. And so it's very controllable. You have much tighter control over dose response. And then finally on durability. So this is, these are rat islets that have been harvested and placed into these capsules, then placed into the mouse. So this is a xenogeneic approach. The mice are immune competent, and they've been treated with STZ, so they're diabetic mice. You can see we've been able to control their blood glucose levels for up to a year. And we've done that with both human-derived beta cells as well as with these with the rat islet cells. In addition, we published a paper in the fall where we did the same thing with non-human primates. So we're able to keep the spheres, the beta cells are encapsulated, happy, healthy, and functional for 4 months in the primates before we terminate the experiment. So what does that allow us to do? There's lots of places you can go. You can think about systemic applications, so where you could deliver continuous steady-state PK like bleeding disorders, lysosomal storage disorders, even some of the rare metabolic diseases. Or you can think about local or regional approaches, so you could think about CNS or ophthalmic. And we also have the opportunity to think about sense and respond opportunities. There's been a few companies talking about that here, which is, you'll notice there's no oncology here, so we're not thinking about oncology, we're thinking about chronic diseases that we can really treat with either a steady-state PK approach or a sense and respond approach where we can create some novel therapies to really create curative therapies for patients. So where are we starting? We wanted to start our pipeline in a couple of areas. One was finding some areas where there was commercial opportunity, but also it is an area where you have a clear regulatory path and clear clinical path. And so we selected hemophilia and lysosomal storage disorders, both of which we feel have significant unmet needs still, and where we feel like we could provide a significant benefit. Finally, in type 1 diabetes, there clearly is a need there and there's an opportunity, but we made a clear choice to only do that if we could find a partner, because that is a very large space, a very challenging space. So our pipeline, today we've got both breadth across therapeutic areas as well as depth within therapeutic areas. And you can see we've disclosed some of the programs we're working on. Our lead will be hemophilia A, which is a target and IND filing end of this year, with multiple other INDs following shortly thereafter. In diabetes, we do have a large partnership with Eli Lilly, which we executed about a year ago. And for us, this is very exciting because it enables us to really target diabetes while at the same time not doing it ourselves, but actually partnering with one of the leading companies in diabetes who knows the diabetes space and can really commercialize and run something through clinical trials. We are accountable for the program up through IND, and so it's very exciting for us to have this as one piece of our platform, but just a piece, and we have a much broader platform beyond that. I think that's it.
Okay, so thank you. So of the three programs of hemophilia, of diabetes, and lysosomal storage disease, can you handicap them? Which one do you think is the easiest, which people think pretty straightforward, or is it unclear?
I think it's a good question. I think you've got different risks across each. So diabetes, we know there's proof of concept in that we know that islet cell transplants from a human donor into a patient can cure that patient. They do have to take immunosuppression and everything else, but we know that's curative, so there is proof of concept there. I think the challenge in diabetes becomes a challenge around making sure you actually get your embryonic stem cell, or in our case, induced pluripotent stem cell, differentiated down into a beta cell. And I think we have a very good approach there, and we're pretty comfortable with it. So that's one risk. On the hemophilia and lysosomal storage disorder side, the beauty of the platform is you're using the same material, everything is the same except we're swapping out the expression cassette. And so that's one of the beauties of the platform. Where your risk then lies really in the different risks that lie within those indications. And I think in the case of hemophilia, you've got clear biomarker. You've got a fairly wide therapeutic window. So we like that one a lot. And in the LSD space, you're dealing with slightly different, perhaps clinical risk, but again, pretty devastating diseases where we believe we can have a real impact.
So do you think your first lysosomal storage disorder attempt would be CNS delivery? It's certainly the unquestionable need.
It is. There's certainly a significant need. Our first will actually be systemic. And we are, notice we did talk about, on the one slide, the ability to deliver to the CNS, and that's something we are looking into heavily because there is a significant unmet need. And we believe that the approach we have should allow us to take an allogeneic approach to actually treating both systemic comorbidities as well as some of the CNS.
Maybe even a silly question, but these are going to be systemic cells, I assume. Any risk in implanting them in the CNS? Is there?
Well, so I think one of the pieces, the beauty of the approach we're taking is it's an allogeneic cell that's in this sphere, in the matrix. And the cells are very controlled, so they can't get out. If you had some type of an event where the cell was, the sphere was crushed or something, you'd probably have other things to worry about. But if that did occur, because of the allogeneic nature, the cells would be rapidly cleared. We see that with MSCs where billions are placed into the circulation. They're gone within a few days.
Okay, I think your CEO would explode out of his chair on this question. But hemophilia, gene therapy is working pretty well. Factors are terrific. What is the opportunity here?
Yeah, that is something we've thought a lot about. And the answer, I think, is pretty easy for us. First, we believe we'll be able to have sustained therapy for years, similar to what gene therapy is promising. I think the second piece is we don't view it as an either-or because you think about it, we can redose whenever we want. The patients, there is no patient that would be ineligible and that no one would have antibodies against our material. So in many ways, we have access to the entire hemophilia population, including those that have been treated with gene therapy. And so for us, it's not an either-or, but it could be an and.
Okay, and in the gene therapy world, I think as it grows up, dosing, lot-to-lot variability, the FDA is starting to get really strict. Maybe you could talk about your control over that parameter. Is that a strength for you, or is it a work in progress?
Absolutely. I think that's one of the beauties of this. Because the cells are engineered ex vivo, the allogeneic cells, we have significant control because we can clonally derive those cells and have a single clone that we then know what its expression profile looks like, both within the spheres and outside of the spheres. And we know what that expression profile will look like in vivo. And so there is much tighter control where you're not throwing in trillions of vectors and seeing 40 to 50-fold different responses in patients, where I think thus far we've seen very tight control when we put in what we believe the right dose is into the animal, and that's what we see what we expect.
And then the last question I have is, what is this? Is this a device? Is this a therapeutic? Are you in a new realm? Do you know?
So we believe we're a new modality. So from a regulatory perspective, we are a combination product, and
Okay.
we have had multiple interactions with both the U.S. and EU regulators, and we are in the U.S. It's within the OTAT within CBER as our most cell and gene therapies. So we are a combination product that is leveraging the power of cells.
I guess I did have one more question. Would you put some sort of kill switch in there in case things go awry? Is that viewed as important?
It's certainly an option. I think in the case of our core programs, we're not using a pluripotent or a multipotent stem cell. It is just it's a fully terminally differentiated cell where the risk of something going awry is very, very low. I think in the pluripotent derived, as we are in, say, diabetes, again, we don't believe we will because, again, we're looking at an allogeneic cell line, and we know allogeneic cells are cleared extremely
Right,
quickly
right.
in a normal immune competent patient because we're not immunosuppressing.
Okay, fantastic. I say, beautiful story. We're out of time, but thank you very much for coming in.